Hi class, this is your instructor, Scholar Huff, and we're now in Chapter 4. This chapter is all about cells. Let us begin. Here you all will state the three principles of the cell theory. They are as follows. All organisms are composed of one or more cells. Cells are the smallest unit of life. And lastly, cells arise from pre-existing cells. In other words, this is a bit of review from back in Chapter 1, where you all learned how this theory came about. In other words, by way of this cell theory, from Hooke, also, we had Schleiden, and then Schwann and Virchow, and we're now here at this cell theory. So just keep in mind that cells are small. You might say, what do you mean by small? Well, if you look in your text to figure 4.1 on page 66, you will see just how small. So in the first picture there, in picture, I guess, numbered 1, you'll see that roughly the area in figure 1 is 20 millimeters. However, as we continue to get zoomed in more and more and more, you'll see that the actual cells themselves are measured in micrometers. So cells are not the same size. And individual marine algal cells can be up to 5 centimeters long, which are huge. However, the cells of our body are typically 5 to 20 micrometers in diameter and way too small for any eye that's naked to see. So it would take anywhere from 100 to 400 cells to span the diameter of a pin, which if you see figure 4.2 in your text on page 67, you're seeing what the diameter of a pin looks like by way of bacteria on that. So you may also ask, why are cells so small? Well, most cells are small because larger cells do not function efficiently. And in the body, it's all about efficiency. So in the center of every cell is the command center. And that is where it must issue orders to all parts of the cell, directing the synthesis of certain enzymes, the entry of ions and molecules from the exterior, and even the assembly of new parts. Hence, these orders must pass through the core to all parts of the cell, and it takes a single molecule a long time to reach the periphery of a large cell. So this is why an organism is composed of relatively small cells has an advantage over being made of one of large cells. So the importance of surface area. So another reason cells are not large is for the advantage of having a greater surface area than volume. So a cell's surface area provides the interior's only opportunity to interact with the environment, as its surface provides the only way for substances to pass into and out of the cell. So as cells grow larger, their interior volume increases much more than their surface area, and as a result, there is far less surface area available to reach each unit of the volume. So in the same way, before airplanes and trains were invented, the size of cities was limited because of the surrounding countryside could not support all the living people in a big city. So only so many roads could be built in the city, and only so many farms were close enough to the city to use them. So larger cells still function quite efficiently because they have the structural features that increase surface area. So cells of the nervous system have long thread-like extensions called axons, extending from more than a meter in length. So then that their interior regions are not far from the surface at any point. For this reason, many body cells are flat and plate-shaped, another structural feature that increases the surface area of a small thing-like projection is called microvilli, which dramatically increases the cell's surface area. So to see cells, what we do is use microscopes. And I'm saying it's because the resolution, which is defined by the minimum distance 2 points can be apart and still be distinguished, it's limited. So the limit of resolution by the human eye is 100 micrometers, and when two objects are closer together than about 100 micrometers, the light reflected from each strikes the same detector cell at the rear of the eye. So the reason we can't see small objects like cells is limited to the resolution of our eye. So we use microscopes. And one way to do such is, well, with a light microscope, using two magnifying lenses and a variety of correcting lenses to achieve a high quality magnification and clarity by magnifying the image and focusing it on the receptor cells in the eye called photoreceptors. So increasing resolution. So light microscopes, even compound ones, are not powerful enough to see many structures within cells. So with this, when two objects are closer than a few hundred nanometers, the light beams reflect from the two images, they start to overlap. So to avoid such, we use a beam of electrons rather than a beam of light. So electrons have a much shorter wavelength, and a microscope employing electron beams has 1,000 times more resolving power than that of a light microscope. So that is why the electron microscope can see much more than merely what can be seen with a light microscope. So if you look closely in your text to figure 4.3, on page 68 it shows you a scale of visibility. And note, on the left-hand side, the giant sequoia, as opposed to a hen's egg. And then from there, things get drastically smaller and smaller and smaller to the paramecium, the human egg, to red blood cells, to bacteria, to a large virus. In that case, it's the human immunodeficiency virus. Then on to the ribosome, the protein, amino acid, and ultimately the hydrogen ion. How small they are. So let's now get to the kinds of cells. There are two kinds of cells. So here you'll describe the interior of a prokaryotic cell. There are two major kinds of cells, the prokaryotic cell and the eukaryotic cell. So prokaryotes have a relatively uniform interior that is not subdivided by internal membranes into separate compartments. They do not have special membrane-bound organelles or a nucleus. So, single-celled organisms. Prokaryotes are the simplest cellular organism. Over 5,000 species are recognized, but doubtless many times that number actually exists and have not yet been described. Although these species are diverse in form, their organization is fundamentally similar. They are single-celled organisms. Their cells are typically small, and I'll say they are smaller than the eukaryotic cell. Their cells are enclosed by a plasma membrane. There are no distinct organelles. The outside of almost all bacteria and archaebacteria is a cell wall composed of different molecules in different groups. And in some bacteria, another layer called the capsule encloses the cell wall. So archaea are extremely diverse and inhabit diverse environments. Bacteria are abundant and play critical roles in many biological processes. And bacteria assume many shapes, such as the sausage or spiral shapes shown in figure 4.4a and b. And they also adhere in masses or chains, as shown in figure 4.4c. But in these cases, the individual cells remain functionally separate from one another. And those can be seen on page 69 in your textbook. The cell interior. If you're able to peer into a prokaryotic cell, you would be struck by its simple organization. The entire interior of the cell is one unit. There is no internal compartments, i.e., no membrane-bound organelles, and little to no internal support. Scattered throughout the cytoplasm of the prokaryotic cells are structures called ribosomes. Those small spherical structures you see on figure 4.5 on page 69. There are, well, before I go there, ribosomes are the site where proteins are made, and they lack a membrane boundary. They're not a membrane-bound organelle. Prokaryotic DNA is found in the nucleoid region of the cell. It is in the cytoplasm. Although the DNA is localized in this region of the cytoplasm, it is not considered a nucleus, and that is because, as you see, the nucleoid region and its associated DNA are not enclosed within an internal membrane. Flagella come next. So some prokaryotes use flagella or a flagellum to move. Flagella are long thread-like structures made of protein fibers that project from the surface of the cell. They are used in locomotion and in feeding, so bacteria can swim at speeds of up to 20 cell diameters per second and rotating their flagella like screws. Some prokaryotic cells contain pili or a pilus, which are short flagella, and the pili or the pilus helps prokaryotic cells attach to appropriate substrates and aid in the exchange of genetic information between cells. So this shows figure 4.5, the organization of the prokaryotic cell. Now to eukaryotic cells. So here you will list the organelles unique to eukaryotic cells and state which of them are not present in plant cells. Eukaryotic cells are much larger and profoundly different from the prokaryotic cells. With the complex interior organization, you can see a cross-sectional diagram of the idealized animal and plant cells on pages 70 and page 71. And as you can see, the interior of the eukaryotic cell is much more complex than that of the prokaryotic cell that you encountered here. For comparison, there. Much more complex. As I continue. The plasma membrane is what encases the semi-fluid matrix called the cytoplasm, which contains within it the nucleus and various cell structures we'll now refer to as being organelles. And in case you have not reviewed yet, or in case you have forgotten, an organelle is a specialized structure within which particular cell processes occur. Each organelle, such as the mitochondrion, has a specific function in the eukaryotic cell. The organelles are anchored at specific locations in the cytoplasm by an internal scaffold of protein fibers called the cytoskeleton. One of the organelles is very visible when these cells are examined with a microscope. Filling the center of the cell, it does, like the pit of a peach. It is known as the nucleus, or nuclei being plural. Inside the nucleus, the DNA is wound tightly around proteins and packaged into compact units called chromosomes. It is the nucleus that gives eukaryotes their name from the Greek words eu and karyon. By way of contrast, the earlier evolving bacteria and archaea are called prokaryotes. So if you examine the organelles in figures 4.6 and 4.7 on pages 70 and 71 respectively, you can see that most of them form separate compartments within the cytoplasm, bounded by their own membranes. So the hallmark of the eukaryotic cell is this compartmentalization with those membrane-bound organelles. This internal compartmentalization is achieved by the extensive endomembrane system that weaves through the cell interior. We'll get on to vesicles here in just a moment. So vesicles, they are a number 7 vesicles are, if you look closely, which includes the lysosomes. So vesicles form closed-off compartments in the cell, allowing different processes to proceed simultaneously without interfering with one another, just as rooms do in a house. Organelles called lysosomes are the recycling centers that have acidic interiors in which old cell organelles are broken down and their component molecules recycled. This acid would be very destructive if released into the cytoplasm. So if you compare figure 4.6 here and figure 4.7 there, you will see the same set of organelles with a few interesting exceptions. Let us begin with those exceptions. For instance, the cells of plants, fungi, and many protists have a strong exterior cell wall, and it is composed of cellulose or chitin fibers, while the cells of animals lack cell walls altogether. All plants and many kinds of protists have chloroplasts within which photosynthesis occurs. No animal or fungal cells contain chloroplasts. Plant cells also contain a large central vacuole. It stores water and plasmodesmata, which are openings in the central cell wall that create cytoplasmic connections between cells. And then centrioles. We'll get to those later, but they are found in animal cells, however not in plant cells or fungal cells. Let's continue on. So now to the plasma membrane, which I just previewed moments ago. The plasma membrane. So encasing all living cells is a delicate sheet of molecules known as the plasma membrane, or the cell membrane, as you may have heard it before. So you will explain how proteins are anchored within the lipid bilayer of a membrane. The fluid mosaic model. So regardless of the kind of cell they enclose, all plasma membranes have the same basic structure of proteins embedded in a sheet of lipids called the fluid mosaic model. The fluid mosaic model refers to the assortment of molecules, i.e., proteins and lipids, that are constantly moving while still maintaining a barrier between the outside environment and the interior or inside of the cell. Phospholipids. So it's the lipid bilayer that forms the foundation of the plasma membrane, composed of modified fat molecules called phospholipids. A phospholipid is a molecule that can be thought of as a polar head with two nonpolar tails. Hopefully you all remember this from our prior chapter when you learned about basic chemistry. This is the phospholipid with the polar hydrophilic glycerol head region and the nonpolar hydrophobic fatty acid tail regions. Lipid bilayers. So imagine what happens when a collection of phospholipid molecules is placed in water. So a structure called a lipid bilayer forms spontaneously. So the long nonpolar tails of the phospholipid molecules are pushed away by the water molecules that are surrounded, shouldered aside as the water molecules seek partners that could form hydrogen bonds. So after much shoving and jostling, every phospholipid molecule ends up with its polar head facing water and its nonpolar tail facing away from water. So the phospholipid molecules form a double layer called a bilayer, i.e. a lipid bilayer. So as you can see in the figure, this figure is known as figure above. Figure above, the watery movements inside and outside the plasma membrane push the nonpolar tails to the interior of the bilayer. Because there are two layers with the tails facing each other, no tails are ever in contact with the water. Thus, the interior of the lipid bilayer is completely nonpolar and it repels any water-soluble molecules that attempt to pass through it, just as a layer of oil stops the passage of a drop of water. And this, of course, is why ducks do not get wet. So cholesterol is also an important part of that plasma membrane. It resides in the interior portion of the bilayer, and the cholesterol is a multi-ringed molecule that affects the fluid nature of the membrane. So although cholesterol is important in maintaining the integrity of the plasma membrane, it can accumulate in blood vessels, forming plaques that lead to cardiovascular disease, i.e. atherosclerosis and even arteriosclerosis. Proteins within the membrane, the second major component of every biological membrane, is a collection of membrane proteins that float within the lipid bilayer. So it's membrane proteins that function as transporters, receptors, and they're there functioning as cell surface markers. So if you look at the figure just shown from your text on page 72 and 73, those purple structures are the proteins. So some proteins pass through the lipid bilayer, providing channels through which molecules and information can pass, while some other membrane proteins are fixed into position, others move about freely. Surface proteins. So many membrane proteins project up from the surface of the plasma membrane, like buoys, often with carbohydrate chains or lipids attached to their tips, like flags. These cell surface proteins act as markers to identify the particular shapes of cells or as beacons to bind specific hormones or proteins to the cell. And lastly, transmembrane proteins. So these extend all the way across the lipid bilayer and can provide passageways for ions and polar molecules like water so they can pass into and out of the cell. So the part of the protein that actually traverses the lipid bilayer is specially constructed of a spatial helix of nonpolar amino acids in red coiled areas of the transmembrane protein you see in the inset. Water responds to these nonpolar amino acids such as it does to nonpolar lipid chains, and as a result, the helical spiral is held to the lipid bilayer interior and then is anchored there by the strong tendency of water to avoid contact with these nonpolar amino acids. So the nucleus. We're now at the cell's control center. So you'll recount two functions of the cell nucleus. Let us begin. So eukaryotic cells have many structural components and organelles in common. If you were to journey far into the interior of one of your cells, you would eventually reach the center of the cell. There you would find cradled within a network of fine filaments, like a ball in a basket, the nucleus, as shown in figure 4.8. The nucleus is the command center and control center of the cell, directing all of its activities. It is also the genetic library where hereditary information is stored. Nuclear membrane. The surface of the nucleus is bounded by a special kind of membrane called the nuclear membrane. The nuclear envelope is actually two membranes, one outside the other, like a sweater or an overshirt. The nuclear envelope acts as a barrier between the nucleus and the cytoplasm, but substances need to pass through the envelope. The exchange of materials through the openings scattered over the surface of this envelope. So nuclear pores are the openings which form when the two membrane layers of one envelope pinch together. So it's that nuclear pore that is not an empty opening. Rather, it has many proteins embedded within it that permit proteins and RNA to pass into and out of the nucleus. Chromosomes. In both prokaryotes and eukaryotes, all hereditary information specifying cell structure and function is encoded in DNA. However, unlike prokaryotic DNA that forms in an enclosed circle, the DNA of eukaryotes is divided into several segments associated with the protein, forming chromosomes. The protein in the chromosomes permit the DNA to wind tightly and condense during cell division. So these condensed chromosomes are readily seen in dividing cells as densely standing rods. After cell division, eukaryotic chromosomes uncoil and fully extend into thread-like strands called chromatin that can no longer be distinguished individually with a light microscope within the nucleus. Once uncoiled, the chromatin is available for protein synthesis. The process begins when RNA copies of genes are made from DNA in the nucleus. The RNA molecules leave the nucleus through the nuclear pores. And as they leave those through the nuclear pores, they enter the cytoplasm where the proteins are synthesized. These proteins carrying out many different functions determine what the cell is like. Now on to ribosomes. So ribosomes are used to make many proteins. So the cell employs a special structure called the ribosome, a kind of platform on which the proteins are built. So ribosomes read the RNA copy of a gene and use that information to direct the construction of a protein. Ribosomes are made of a special form of RNA called ribosomal RNA, or rRNA, that is bound up within a complex of several dozen different proteins. Ribosomal subunits are assembled in a region within the nucleus called the nucleolus. The endomembrane system is next. We'll begin with the endoplasmic reticulum. The extensive system of internal membranes is called the endoplasmic reticulum, often abbreviated as ER. Looking at the sheets of the membrane weaving through the interior of the cell, you can see how the ER got its name. Please see figure 4.9 on page 76. The endoplasmic reticulum creates a series of channels and interconnections. It also isolates some spaces of its membrane enclosed sacs called vesicles. The surface of the ER is the place where the cell makes proteins intended for export, such as enzymes secreted from the cell surface. The surface of those regions of the ER is devoted to the synthesis of such transported proteins that is heavily studded with ribosomes and appears pebbly or just studded, like the surface of sandpaper. When seen through an electron microscope, it appears quite readily. For this reason, these regions are called rough endoplasmic reticulum. Regions in the rough ER are relatively scarce and are correspondingly called rough ER. Regions in which ribosomes are relatively scarce are called smooth ER. So there is more endoplasmic reticulum with ribosomes, i.e., rough endoplasmic reticulum, compared to those regions that are scarcely studded with ribosomes, and that is known as the smooth endoplasmic reticulum. So having finished both of those, we'll move then nextly to the Golgi complex, or the Golgi apparatus as some may have taught you. So this shows the area at the top with those blue ribosomes being the rough ER, and the area near the bottom that has relatively few or even no ribosomes being the smooth ER. The Golgi apparatus or Golgi bodies. So you explain how here the Golgi bodies ensure correct delivery of substances made in the ER. As new molecules are made on the surface of the ER, they are passed from the ER to flattened stacks of membranous sacs called Golgi bodies. They look like pancakes stacked one on top of another. Golgi bodies function in the collection, the packaging, and distribution of molecules manufactured in the cell. So scattered through the cytoplasm, Golgi bodies are collectively referred to as Golgi complex. The rough ER and smooth ER, and even the Golgi work together as the endomembrane transport system in the cell. So if you look at figure 4.11, it walks you through the path that the molecules take from ER through Golgi and out to their final destinations. That photo is shown on page 77. Proteins and lipids are manufactured in the endoplasmic reticulum. In their membranes, they are then transported through the channels of the ER and are packaged into transport vesicles that bud off the endoplasmic reticulum, and then those vesicles will fuse with the membrane of the Golgi bodies, dumping their contents into the Golgi complex. Within the Golgi bodies, the molecules may take one or more paths indicated by the branching arrows in figure 4.11, and then many of these molecules become tagged with carbohydrates, and then the molecules collect at the ends of the membranous folds of the Golgi bodies. So these folds are given a special name, cisternae. So vesicles that pinch off from the cisternae carry the molecules to different compartments of the cell and or to the inner surface of the plasma membrane, where the molecules will be secreted and released to the outside. Lysosomes are coming in next. So this is Golgi, the Golgi complex. So this is figure 4.11 that shows how the endomembrane system works entirely, from the nucleus to the smoothie yard, to the roughy yard, to the Golgi complex, to the vesicle, and then ultimately to that vesicle fusing with the plasma membrane and then secreting or its product. Lysosomes. So they arise from the Golgi complex, and they contain a concentrated mix of powerful enzymes manufactured in the rough endoplasmic reticulum that break down macromolecules. Hint, hint, carbohydrates, lipids, proteins. Lysosomes are the recycling centers of the cell, breaking down failing organelles and other structures within cells, so digesting those worn out cell components and recycling proteins and other materials of the old parts. Next up are vacuoles. So the interior of many plant and protocells contain membrane-bound storage compartments called vacuoles. The center of the plant cell is shown in figure 4.12 on page 77. As in all plant cells, contains a large, apparently empty space called the central vacuole. The vacuole is not really empty. It contains large amounts of water and other materials such as sugars, ions, and pigments. The central vacuole functions as a storage center for these important substances. Organelles harvest energy. So here you'll differentiate between the mitochondrion and chloroplasts. So the mitochondrion, it says, powerhouses of the cells. Eukaryotic organisms extract energy from organic molecules in a complex series of chemical reactions called cellular respiration, which takes place only in the mitochondria. So mitochondria, which is plural, mitochondria in the singular, are sausage-shaped organelles about the size of a bacterial cell. Mitochondria are bound by two membranes. The outer membrane, shown partially cut away in figure 4.13 on page 78, and then we also have the inner membrane. So the outer membrane is smooth and apparently derives from the plasma membrane of the host cell that first took up the bacterium years ago. The inner membrane, apparently, the plasma membrane of the bacterium that gave rise to the mitochondrion, is bent into numerous folds called cristae. The cristae resemble the folded plasma membranes in various groups of bacteria. The cutaway view of the figure shows how the cristae partition of the mitochondrion into two compartments, an inner matrix and an outer compartment, the intermembrane space, that outer compartment. As you will learn later, this architecture is critical to successfully carrying out cellular respiration. So mitochondria have a circular molecule of DNA called the mitochondrial DNA that closely resembles the circular DNA molecule of a bacterium. So it's on this mitochondrial DNA that several genes that produce some of the proteins essential for cellular respiration. Chloroplasts, energy capturing centers. So some organisms are able to capture energy from sunlight and use it to power the production of sugars in a process called photosynthesis. All photosynthesis in plants and algae takes place within another bacterial-like organelle, the chloroplast, seen in figure 4.14. This is on page 79. There is strong evidence that chloroplasts, like mitochondria, were derived from bacteria by mutually sharing an arrangement called symbiosis. A chloroplast is bounded like a mitochondrion by two membranes, the inner derived from the original bacterium and the outer resembling the host cell's endoplasmic reticulum. Chloroplasts are larger than mitochondria and have more complex organization. Inside chloroplasts, another series of membrane is fused to form stacks of closed vesicles called thylakoids. The energy harvesting reactions of photosynthesis take place within the thylakoids. The thylakoids are stacked on top of one another to form a column called a granum, or grana being plural. The interior of a chloroplast is bathed with a semi-liquid substance called the stroma. So like mitochondria, chloroplasts have a circular DNA molecule. The DNA contains many of the genes coding for the proteins necessary to carry out photosynthesis. Plant cells can contain from 1 to several hundred chloroplasts, depending upon the species. Neither mitochondria nor chloroplasts can be grown in a cell-free culture. They are totally dependent on the cells within which they occur. So the endosymbiotic theory, or the theory of endosymbiosis. Symbiosis is a close integrated relationship between organisms of different species that live together. The theory of endosymbiosis proposes that some of today's eukaryotic organelles evolved from symbiosis in which one cell of the prokaryotic species was engulfed by and lived inside the cell of another species of prokaryote that was a precursor to eukaryotes. So figure 4.14 on page 29 shows this. Many cells take up food and other substances through endocytosis, a process by which the plasma membrane of a cell wraps around the substance, enclosing it within a vesicle inside the cell. According to the endosymbiont theory, the engulfed prokaryotes provided their hosts with certain advantages associated with special metabolic abilities. Two key eukaryotic organelles just described are believed to be descendants of the endosymbiotic prokaryotes: mitochondria, which are thought to have originated as bacteria, capable of carrying out oxidative metabolism, and chloroplasts, which apparently arose from photosynthetic bacteria. So the endosymbiont theory is supported by a wealth of evidence. Both mitochondria and chloroplasts are surrounded by two membranes. The inner membrane probably evolved from the plasma membrane of the engulfed bacterium, while the outer membrane is probably derived from the plasma membrane or the endoplasmic reticulum of the host cell. So the mitochondria are about the size of most bacteria, and the cristae formed by the inner membranes resemble the folded membranes of various groups of bacteria. So mitochondrial ribosomes are also similar to the bacterial ribosomes in size and structure. Both mitochondria and chloroplasts contain circular molecules of DNA, similar to those in bacteria. And then finally, mitochondria divide by simple fission, splitting into two, just as bacterial cells do. And they apparently replicate and partition their DNA in much the same way bacteria do. During the 1.5 billion years in which chloroplasts and mitochondria have existed in eukaryotic cells, most of their genes have been transferred into the chromosomes of host cells, but chloroplasts and mitochondria still have some of their original genes. This is the endosymbiont theory. So now we'll get to the cytoskeleton, the interior framework of the cell. So here you'll describe the protein fibers of the cytoskeleton. So if you were to shrink down and enter into the interior of a eukaryotic cell, your view would be similar to what you see in the illustration shown here. This illustration can be seen on page 18. It looks a bit like this. So we have microfilaments or actin filaments. We have microtubules. We also have intermediate filaments. And I'll start with the microfilaments quickly. So the microfilaments are by way of the protein actin. You'll learn more about actin when you learn about muscle. Microtubules, composed of tubulin, are a bit different in their structure. So whereas the microfilaments have about 7 nanometers in diameter, the microtubules have about 25 nanometers in diameter. And the microtubules are made by way of the protein tubulin. Intermediate filaments are a bit different. So here the intermediate filaments, they twist into bundles, into cables, and once they are formed, they usually do not break down. And that is all you all will have for me on microfilaments, microtubules, and those intermediate filaments. But one thing I will say about microtubules, the ones that group together, they can either form flagella or cilia. So quickly I'll mention the two. Some eukaryotic cells contain flagella. And if you look at figure 4.16a, you'll see that flagella are there, and what they're used for is movement. And then with cilia do not differ from flagella in their structure, but they differ in their function. So cilia are typically shorter, and in humans, dense massive cilia project to line our breathing tube by way of the trachea, and they move mucus and dust particles out of the respiratory tract and the throat. More specifically, I would say that flagellum are used for movement as opposed to the moving material. So in humans, we have a single flagellum on each sperm cell that propels the sperm cell in a swimming motion. Now on to cellular transport by way of diffusion and osmosis. So here you explain why diffusion occurs down a concentration gradient rather than up. For cells to survive, nutrients, water, and other materials must pass into the cell, and waste materials must be eliminated. So all of this moving back and forth across the cell's plasma membrane occurs in one of three ways. Water and other substances diffuse through the membrane. Food particles are engulfed by the plasma membrane folding around them, or proteins in the membrane acting as doors to admit certain molecules only. So we'll begin with diffusion. So how a molecule moves is just where it goes. It's totally random, like shaking marbles in a cup. So if two kinds of molecules are added together, they soon mix. So the random motion of molecules always tends to produce a uniform mixture or a uniform mix. And because the substances move from regions where the concentration is high to regions where the concentration is lower, i.e. down the concentration gradient. So now the question is, how does a molecule know what direction to move? The molecule doesn't know. It doesn't know a thing. So a molecule, as it moves by way of diffusion, it is equally likely to move in any direction and is constantly changing course in random ways. So there are simply more molecules able to move from where they are common than from where they are scarce. This mixing process is known as diffusion. So diffusion is the net movement of molecules down a concentration gradient towards a region of lower concentration as a result of random motion. For example, a lump of sugar dropped into a beaker of water, and you can see this essential biological process 4a on the left-hand side of page 82. So that lump of sugar dropped into a beaker of water will break apart into individual sugar molecules that will move about randomly. However, they will tend to travel away from the area of high concentration to an area of lower concentration. And eventually the substance will achieve a state of equilibrium where there is no net movement toward any particular direction, as shown as in panel 4 or figure number 4. So the individual molecules of the substance are still in motion, but there is no net change in direction. Osmosis. As in diffusion, water moves into and out of cells by a form of diffusion called osmosis. As you can see here, only certain substances undergo diffusion across the plasma membrane, and there are specific examples provided to you all in their class. So, as in diffusion, water passes across the cell membrane down its concentration gradient, a process called osmosis. You will see this essential biological process 4b on page 83 on the right hand side of your page. So, to understand how water moves into and out of a cell, let's focus on how the water molecules are already present inside the cell. So many of those water molecules are interacting with sugars, proteins, and other polar molecules inside. And remember, water is a very polar molecule itself, and it rarely interacts with other polar molecules instead of freely moving about. A shell of water molecules remains clustered around each polar molecule inside the cell. As a result, a water molecule coming into the cell by random motion may not be free to come out again. So the addition of a polar solute reduces the number of free water molecules on the right side of the beaker you all see on page 83. So because the outside of the cell on the left has more unbound water molecules, water moves by diffusion into the cell on the right from a region of hypotonicity to a region of hypertonicity, which I'll explain in just a second. So aquaporins, so ions and polar molecules cannot pass the very nonpolar environment found in the lipid core of the cell membrane bilayer. However, the movement of water molecules, which are very polar, is not blocked. Water diffuses freely across the plasma membrane, and that happens by way of the water molecules passing through small channels called aquaporins. Aquaporins traverse the membrane. So while the water can cross the membrane very slowly on its own, aquaporins allow the water to move much quicker. These water channels are very selective, even blocking the passage of protons, which are hydrogen ions, which are smaller than water molecules. So a cluster of positively charged amino acids that line the pore repel protons, which are also positively charged. Now to osmotic concentration. So the concentration of all solutes dissolved in a solution, i.e. solutes, is called osmotic concentration of a solution. So if the osmotic concentration of the two solutions are equal, the solutions are isotonic. If two solutions have unequal osmotic concentrations, the solution with the higher solute concentration is said to be hypertonic, i.e. higher solute concentration, hypertonic, as you see in plate 2. Right-hand side, hypertonic. And lastly, the solution with the lower one is hypotonic. So the lower concentration of solutes, hypo or hypotonic, such as on the left in plate 2, as you see it. So movement of water into a cell by osmosis creates pressure, called osmotic pressure, which can cause a cell to swell and burst, which you can see in figure 4.17 at the bottom of page 83. Most animals cannot withstand osmotic pressures unless their plasma membranes are braced to resist the swelling. If placed in pure water, they soon burst like an overinflated balloon. That is why cells of so many kinds of organisms have cell walls to stiffen their exteriors. In fact, this osmotic pressure, called turgor pressure in plants, is important for plants to maintain their shape. Without adequate water inside the cells, the plants wilt, and in animals, the fluids bathing cells may have many polar molecules dissolved in them. So, as that happens, that makes them isotonic. So again, in animals, the fluids bathing cells may have as many polar molecules dissolved in them as they do inside their cells, making them iso or equal, meaning isotonic. So the problem does not arise. So, having reviewed these, let's get to this. You're looking at the behavior of animal cells and plant cells in those three different types of solutions. In the center, top center, you're seeing the isotonic solution. Animal cells are normal in isotonic solutions. The solute concentration is the same both inside that cell and outside the cell. There's no net difference in the amount, no net difference in the amount of water, or no net difference in the amount of volume of water entering or leaving that cell. On the left-hand side, the hypertonic solution. So since the hypotonic cell is in a hypertonic solution, it will shrivel, it will lose water, and it will shrivel up just as you see, from hypo to hypertonic solution. On the right-hand side, you see the hypotonic solution. So animal cells will swell, they will burst or lyse in hypotonic solution. It's because the hypertonic cell to that hypotonic solution. Hence, the water will enter the cell from a hypotonic solution to that hypertonic cell. The cell will swell and burst and or lyse. At the bottom, plant cells are normal or turgid in hypotonic solutions. In an isotonic solution, in the center, bottom, plant cells will be flaccid or wilt. And in a hypertonic solution, the plasma membrane will separate from the cell wall of that animal cell. So animal cells are not normal in hypertonic solutions. So let's go to bulk transport. With bulk transport, this is going to be the movement of large, I guess I'll say liquids or large materials. So here we have endocytosis. So not all movement of materials into and out of cells happens through a channel. The cells themselves may take in that food or liquid. They do this by extending their membranes outward toward those particles. So the membrane engulfs the particle and forms a vesicle, a membrane-bound sac around it, and this is called endocytosis. And there are two types of endocytosis. So if the material the cell takes in is particulate, or at least not a liquid, or some other fragment or other organic matter, the process is called phagocytosis. If the material is liquid or a substance that is dissolved in liquid, then it is referred to as being pinocytosis. So I call phagocytosis cell eating and pinocytosis cell drinking. So pinocytosis is common among animal cells. Mammalian egg cells, for example, they nurse the surrounding cells nearby by secreting nutrients that maturing egg cells take up by pinocytosis. So virtually all eukaryotic cells constantly carry out these kinds of endocytosis, trapping particles and extracellular fluids in vesicles and ingesting them. So endocytosis rates, they vary, but they can be surprisingly high, with white blood cells ingesting 25% of their volume each hour. Next up is exocytosis. So this is the reverse of endocytosis. So exocytosis is the discharge of material from vesicles at the cell surface. So this is released from the cell. So as you can see in the figure there, figure 4.19, the cell is releasing by way of that secretory vesicle, the secretory product. So now we're to selectively permeability. Almost at the end of the chapter, you all. So here in our last section, you will distinguish between selective diffusion and facilitated diffusion. So from the point of efficiency, the problem with endocytosis is that it's expensive to carry out, and the cell must make and move a lot of membrane. So endocytosis is not picky. In pinocytosis, particularly engulfing liquid does not allow the cell to choose which molecules come in. This is solved by, the problem I'm referring to is solved by using proteins in the plasma membrane as channels to carry proteins to pass molecules into and out of the cell, because each kind of transport allows the passage of only a certain kind of molecule. So the cell can control what enters and leaves by way of selective permeability. Let's get to selective diffusion. So some transport proteins are channels that act like open doors. As long as the molecule fits the channel, it is free to pass through in either direction. Diffusion tends to equalize the concentration of such molecules on both sides of the membrane, with the molecules moving toward the side where they are the scarcest. This mechanism is called selective diffusion. One class of selectivity opened channels consists of ion channels, which are pores that span the membrane. So ions that fit the pore can diffuse through in either direction, such as ion channels play a role in signaling by the nervous system, such as the sodium ion channels. Sodium ion channels allow sodium to diffuse into the cell, and that allows the sodium to also then after leave the cell. Facilitated diffusion. Most diffusion occurs through use of special carrier proteins. These proteins bind only to certain kinds of molecules, such as a particular sugar, or an amino acid, or even an ion. So the molecule physically binds to the carrier on that side of the membrane and is released to the other side. So as I said a moment ago, sodium ion channels. So with that, the direction of the molecule's net movement depends on its concentration across the membrane. If the concentration is greater outside the cell, the molecule is more likely to bind to the carrier on the extracellular side and then move directly in, as you can see in Biological Process 4C on the right side of page 85. So if the concentration of the molecule is greater inside the cell, the net movement will be from the inside to outside. Thus, the net movement is always going to occur from a high concentration to low concentration, just as it does in simple diffusion. But the process is facilitated by the carriers. So for this reason, the mechanism of transport is given a special name, facilitated diffusion. So a characteristic feature of the transport by carrier proteins is that its rate can be saturated. So if the concentration of a substance is progressively increased, the rate of transport of the substance increases up to a certain point, then levels off. So there are a limited number of carrier proteins in the membrane, and when the concentration of the transport substance is raised enough, all the carriers will be in use. So the transport system is said to be saturated. Active transport. So here you'll describe the operation of the sodium-potassium pump. You will see this as essential biological process 4D as in dog on page 87. So other carrier proteins through the plasma membrane are closed doors. These proteins open only when energy is provided. They are designed to enable the cell to maintain high or low concentrations of certain molecules, much more or less than exists outside the cell. Like motor-driven turnstiles, these transport proteins operate to move a certain substance up and its concentration gradient. I repeat, these transport proteins operate to move a certain substance up its concentration gradient, or against the concentration gradient, if that helps better. So the operation of these one-way energy-requiring proteins results in active transport, the movement of molecules across the membrane to a region of higher concentration using energy. So you might think the plasma membrane possesses all sorts of active transport proteins for the transport of sugars, amino acids, and other molecules, but in fact, most of the active transport in cells is carried out by one kind of transporter, the sodium-potassium pump. So the sodium-potassium pump is the most important active transport protein is the sodium-potassium pump. So it expends metabolic energy to actively pump sodium ions out of cells and potassium ions into cells. More than 1/3 of all energy expended by your body's cells is spent driving sodium and potassium pump carrier proteins. This energy is derived from adenosine triphosphate, or ATP, a molecule we will learn more about in a subsequent chapter called 5, our next chapter. So the transportation of two different ions in opposite directions happens because the energy to cause a change in the shape of the protein carrier. So the panels to the right, meaning the essential biological process 4D, walks you through one cycle of the pump. So each transport protein can move over 300 sodium ions per second, working at full tilt. As a result, there are far fewer sodiums inside the cell. Coupled transport. So the plasma membranes of many cells are studded with facilitated diffusion transport proteins, which offer a path for sodium ions that have been pumped out of the sodium-potassium pump that diffuse back in. There's the catch, however. These transport proteins require the sodium ions have to have a partner in order to pass. This is why these are called coupled transport proteins. Coupled transport proteins won't let sodium ions cross unless another molecule tags along, crossing hand in hand with the sodium ion. In some cases, the partner molecule is a sugar, in others, an amino acid, and in another, a molecule. Because the concentration gradient for sodium is so large, many sodium ions are trying to get back in. This diffusion pressure drags in the partner molecules as well, even if they're already in high concentration in the cell. In this way, sugars and other actively transported molecules into the cell via special coupled transport protein channels. So once again, class, this has been chapter, what is called 4. Make sure you put the chapter to work, class. Also, make sure you go to retracing the learning path. So there will be 3 sodiums pumped out of the cell and 2 potassium ions pumped into the cell using energy in the form of ATP. So this has been your instructor, Scholar Huff. Please review this well and prepare well for your test. Thank you all for listening.